talk in Sydney before. So it's always amazing to come to this beautiful city and this beautiful campus to share knowledge and expertise. So yeah, as Sheila mentioned, a lot of our work, oops, next slide, oops, no, hang on. Just have to get a new PhD to learn how to advance the slide. Okay, click there, good. So I guess a lot of our time, as probably for most data scientists in the room and those online, is spent with our head in this sort of data matrix. So what is in the matrix? What kind of features can we measure using cool genomic technology? Is it gene expression? Is it chromatin accessibility? And are we measuring it at bulk level or at a single cell level, or at today's talk at a spatial level? So just to give you a sense of the types of work my lab does, at the WEHI, we have a lot of bioconductor packages that support constructing the data matrix. So tools like SCPipe are about, you've got your raw sequencing reads from a single cell experiment. There's lots of different single cell protocols out there, but you can use this tool to create a feature matrix with genes in the rows. Oh, I have some squigglers. Feel free to annotate the slides as we go at home. If I make any mistakes, you can put some red lines through, particularly if there's typos, that would be helpful. Another package, Flames, is about, how do you take the raw data from a long read single cell experiment and again, make inference on that? Because I guess transcript level data on single cells is usually not there. It's usually 3' gene count level data. And tools like SpatialBench is like, if we have lots of different single cell data sets and we want to use them for benchmarking across lots of different steps in the analysis pipeline, how can we do that at scale and have really complicated analysis pipelines with multi-methods and multi-data inputs? Other things are about how can you add value to this matrix, maybe capture intron signal, which is often currently sort of ignored. So there's packages, superintronic for that. How do you annotate your data matrix? Once you've done your analysis to try and prioritize the features of interest, can you interactively explore that so that the biologists that you send your reports to can, in their R Markdown, have an easy point-and-click solution to find their favorite genes rather than email you saying, where is this gene and where's that gene? Can you highlight it in the plots? Like, well, tools like Glimmer allow people to do that themselves. And then in the long read space, methylation is a cool signal you can get from your beautiful long reads of technologies like nanopore sequencing, restrander for doing some QC on long read RNA-seq. And we've got a database called Long Read Tools, which sort of collates all the different methods that are out there being developed to support long read analysis, be it DNA or RNA-seq or other applications. And if I think back to microarrays and then bulk RNA-seq, there's obviously tools for, again, prioritizing the rows in your data matrix to hopefully send to your collaborators so that they can validate things in the lab. And so software like Limmer has lots of methods for that, and EXE for kind of gene set enrichment analysis, where you apply lots of different methods to your data to come up with some consensus approach. And I guess the goal of having a sharper data matrix with more features in the rows is getting away from the kind of low resolution view that maybe you're used to if you played video games maybe 10, 15 years ago. Mario used to look quite pixelated, but now with more pixels on the screen, it looks quite sharp. And you can definitely, tell his arms, they're more well-defined, and his face, his mustache, his eyes, you can kind of get a better sense of how he really looks. And of course, in genomics, we're really lucky. And I really love this leaderboard of Nature methods of the year, where they go and they review, the latest cool papers across all the different technologies that are available to researchers and try and rank them, basically announce, okay, Nature Method of the Year 2007 is second generation sequencing. And so, yeah, lots of cool technologies, be it microscopy, lattice light sheet, methods for measuring transcriptome, epitranscriptome, all of these are adding extra features to your data matrix. And of course, we need tools that are going to make good sense of those. So it's nice to look at this timeline. So for me, it's like, well, second generation sequencing was 2007 method of the year. I don't think I saw that sort of data for quite a few years after that. So there's kind of a bit of a lag between this leaderboard picking it up as the method of the year and maybe you getting data, or maybe vendors getting involved and having a really easy workflow so that people can routinely generate this, but you can kind of be sure that if it's in the list, soon enough, you're going to see data from it. And of course, we need to be across how to analyze that data when it hits our desks. So today's talk is Nature Method of the Year 2020, Spatially Resolved Transcriptomics. So that's the focus of our benchmarking today. 2022 Method of the Year was long read sequencing. And I believe a former student, Shu Dong, gave a seminar in this series a few weeks ago about our long bench benchmarking data set, which was really to look at how long read bulk RNA-seq methods compare with existing tools to develop a pipeline for that sort of data. So I guess with all of these platforms, I guess life can get quite complicated. So, you know, for every new sequencing-based platform or spatial platform, there probably won't be one, there might be multiple. And then for every different analysis task for that platform, there might be a multitude of different tools that you can use to do that analysis. So I guess the goal of methods benchmarking and creating reference data sets is to try and simplify life from having a fairly complicated all-in-one pocket tool to something that's maybe a bit more refined. Okay, for this sort of data, we use this followed by this, and we don't have to try and explore all of the different options that are available to us, which of course grows and grows over time. If you want to, you know, track the single cell universe of tools, the scRNA tools database is a good one, and there's various ones for spatial omics as well. And I mentioned previously the long read tools database to track tools that make that type of analysis easier. So yeah, and ideally when we apply these tools to our data, we want to have, you know, results that our biologists will want to spend time, our collaborators following up, and not basically give them a random list or a list full of false positives, which will be, you know, surprising, but maybe not worth pursuing further. So no surprising results. Well, surprising, but not too surprising. So I wanted to give you a bit of a sense of, you know, how do we make these benchmarking data sets? So, you know, for microarray, bulk RNA-seq, single cell, you know, people from time to time design these bespoke experiments which have inbuilt ground truth, and there's particular ways that people create these reference samples in the lab to make them, you know, have some idea of which things are going up, which things are going down, or which things are not changing. So I guess this is the bit where I throw to the audience. Does anyone want to guess? So every element is in this picture. And because it's lunchtime and people might be teaching, all of these are non-alcoholic things that we're mixing to make a delicious thing we can enjoy just after lunch. It's not going to put us to sleep for our afternoon lecture or slurring if we are giving the lecture. So does anyone want to have a guess? It's 3 elements and that they're in the picture. And if you're on Zoom, you can probably put in the chat if you want to guess. Anyone?
Easy, fast.
Fast, okay. Yes, you don't want to wait too long for your cocktail. But no, that's not what I was looking for. Sorry. There's no time, there's no stopwatch in this picture. So that's not... So what I'm asking is, yeah, how would you make your sample? If you had to make a cocktail, a biological cocktail, what might you put in it? Yeah.
Instructions.
Instructions, yes. Recipe, yes. This is good. So I guess if you follow a recipe, you're going to know what you put in your mix, what you put in your reference sample, and hopefully you can read that back out with the technology that you applied to generate the data. So yeah, recipe is definitely a good answer. I guess the kind of terms that I think of that are depicted here when we make a reference sample, it's worked really well for bulk RNA-seq and single cell RNA-seq as spike-ins. So often there's particular RNA molecules that are a bit different from the rest of your sample that you put in, and you know they have particular concentrations. So ideally you want to read back that concentration. So you can see there's 3 olives on a spike. So there's the spike. Another element is mixture. So I guess it's like following a recipe is how do we mix, you know, different combinations of single cells, different species of RNA, if it's bulk RNA, in certain known concentrations, so that you say, right, we've got a pure sample, we've got a 50-50 mixture. So obviously, you know, if we compare and we've got another pure sample, we can compare the pure things where things will be most different, and then the intermediate mixtures will be sort of in between. So for those sorts of setups, basically every gene can change at different levels depending on their abundance in the different pure samples. And then hopefully you can read it across the mixture series. And hopefully the method will quantify that change in expression really accurately for you. The final thing, maybe you wouldn't do, the concept of our dilution. So it would be like, you could have, is there a shot glass there? Well, you could pour in neat, or you could then make a 50-50. And then, so then again, that's another situation where everything across the series of samples you might make for your reference is going to go down systematically, depending on how you make it. So those are the three elements. Now, does this help us in spatial? Not really. I'm still not really sure how you would make a cocktail of cells that have a specific spatial, location or orientation. So all of these sorts of strategies are great when you've got molecules floating around that you can just sample and then mix in different contributions. So for spatial, we have a bit of a challenge. So how do we control gene expression in a spatial context? Well, I guess the answer is we can't really. But of course, a lot of people have these go-to reference samples where they really have studied, the anatomy, what sort of features should be there, what sort of cells should be where, and we can use that biological ground truth. I mean, another thing that one can do is just by varying the types of tissue. So for example, if you get tissue from a male sample and tissue from a female sample and they're healthy tissue, then the only things that will change might be sex-specific genes. And similarly, if you have tissue from genes where you sample where you knock out a gene and you've got your control, again, there's sort of the particular perturbations you do to samples, you can try and use that. And again, will the spatial technology read that back to us or not? So that's what we'll be doing today. We'll be using some reference tissues that are coming from a real biological entity. So the first part of the talk is about a project we call SpatialBench. And this is focused on 10x Visium spatial transcriptomic technology. So the problem was presented to us by our colleagues who run the genomics lab, which were like, we're consistently getting approaches from different vendors who have these commercially available platforms. And, which one should we buy? Should we buy them all? Well, we can't afford them all. Should we focus on, which one? So the idea here was from the array of commercially available spatial profiling technologies, which ones might work on different types of tissues. So the 6 options here that were originally planned for this study were Visium, Stomix, Curio, which are sequencing-based methods, and Merscope, Xenium, and Cosmix for the imaging-based platforms. And of course, more methods will come online. So ideally, if you design a study, you could try and choose a reference tissue that you can add to over time so that it can be useful to keep the results and data sets as contemporary as possible and as useful as possible. So as well as there being lots of different commercially available platforms for reading back spatial transcriptomic information, there's obviously lots of analysis methods that are existing or being developed for spatial technology. So spatially aware versions of all the usual things you might want to do with single cell data, be it clustering, deconvolution is a type of analysis particular for spatial data, and so on and so forth. So yeah, there's often, again, as the Pocket tool before showed, there's lots of options for that as well. So we've got plenty of things to compare, and having access to a benchmarking data set to allow this to be done systematically could be of value. So that was the goal of this study. So the tissue chosen here, typical benchmarking studies up to now are usually focused on brain, but we chose spleen because it was available. And I guess coming from an institute with lots of immunologists, there's a lot of interest in having lots of different immune cells. How do they look like in different organs? So spleen is a very rich bag of immune cells. So the particular model here is the spleens have been challenged with malaria, and then they've been drug cured. And so basically this means that particular regions of the spleen have been activated and have been more observable than they ordinarily would be. And these fall into zones such as germinal centers, which are the little regions there shown in red. You can see on the image on the left. What else we have? Naive B cells that sort of surround these germinal centers, and then patches of T cells, which are kind of immediately adjacent to those as well. And then lots of infrastructure like cells that sit between it and other blood cells that you expect in these different regions as well. So hopefully we can read back this kind of splodgy pattern where there's germinal centers for sure, because we've challenged these mice with malaria, and T cells and B cells and other little patches of cells across this tissue. So we have also samples that come from different genotype animals. So we've got wild type spleens, also ones that are knockout and control for a particular transcription factor called TBET that the collaborators were interested in. And samples from different biological sexes prepared across all the different ways that you can ideally prepare these samples. So you can freeze them fresh, so fresh frozen or OCT. You can put them in FFPE to preserve them, which is a common way of preserving tissues, and then across as many platforms as we can have. And also have reference single cell data so that, again, for all the kind of deconvolution and other annotation tasks, you can use that as well. So when we make RNA cocktails with all the spike-ins, it's pretty easy to sort of make the reference sample, make enough of it, make more of it if you need to. For this sort of benchmarking, actually getting good data was the first challenge. There's quite a steep learning curve being able to generate good data. So this first challenge here shows early experiments which use Visium technology, where if you look really carefully, there's bubbles of varying sizes, really tiny bubbles on the left, medium-sized bubbles in the middle, and really large bubbles on the right, which apparently is bluing buffer bubbling problem, which doesn't really mean a whole lot to me. But if you get that wrong, you get bubbles of varying sizes, which are not going to help you read back the different regions of the spleen, because you have these weird bubbles instead. So this was something that could be resolved through working with the technical support people from Visium, which is great. There were a lot of issues getting data for stomics, just for the fact of, again, getting the analysis pipeline to work was challenging. So you can see here an example of the spleen is a really densely packed tissue. So maybe that's another challenge. So you can see the default segmentation done here is perhaps missing a lot of cells. And then if you do something custom, you can get more cells per region, and they look like each little yellow circle surrounds a cell, so that you're doing a better job there by making a custom version of the segmentation. Another technology we tried was an issue with getting the image in focus and having some gel lifting, which basically means all the bit that's lifted, you don't really get any signal from. So again, edge artifacts related to how you place the tissue, how you cover the tissue. And so, yeah, data there was not forthcoming on the first attempt either. But we're lucky the people in the lab go back are still optimizing many of these things. So I think there's hope that there'll be data from other platforms soon. But in the meantime, what did work really well after we got through the bluing buffer bubbling problems was there was lots of Visium data across the range of different tissue genotypes, male and female samples, and across all the different ways of preparing these tissues, from fresh frozen or OCT, as denoted in this table, with or without the special instrument that you get from 10X, which is a cytosist, which is denoted by CA on these slides, which helps you position the tissue on the slide and basically get the most data you can, highest quality data you can. And you can see there, depending on whether you use, whether you have FFP data or use the cytosist approach or not, there's different ways of actually making the library. So some methods are probe-based, where you're only going to read back the particular region of the gene that has been targeted in the assay, whereas the fresh frozen, you can do a poly-A, which doesn't rely on probes. So there's a range of things going on in this experiment, and having this sort of really deep data set across Visium means we can really road test, well, what difference does sample handling method have on our data quality? Does it matter if you have probes or not? How does that impact things? And of course, we have a single cell reference sample as well that we can use for different tasks, and compare that to how well things work if you just use a public reference data set, for example. So the workflow is as follows. Most of the first part here is what people in the lab do to prepare the sample, make the library, sequence the library, and then it comes down to us to go, right, we need to make run Space Ranger to make our data matrix, and then do downstream analysis using other tools to deal with the quality control, to make sure we're only keeping spots that are actually covered by a bit of tissue, making sure we're only keeping spots that have a reasonable number of reads in terms of library size or number of features detected, and basically clean up the data using tools like SCADA and SCRAN, or methods in there that are also applicable to regular single cell data, although here, of course, we're dealing at the level of a spot. Feature selection is important. We want to choose which features go into the rest of our analysis, so we can either go for highly variable genes or spatially variable genes, and there's methods in SCRAN and NSVG for doing that. And then dimensionality reduction and downstream analysis, which encompasses clustering, marker gene detection, and annotating those, and other steps as well. Cell type deconvolution and visualization. And so for all of these, we've used open source bioconductor tools, in particular NNSVG for the spatially variable gene analysis. And what's nice about that is if you have multiple samples, as we have for each and every condition that we have, we can do that and get a consensus spatially variable gene list, which is nice. Likewise, for clustering, methods like ISC-MEB allows you to cluster kind of across replicate samples, which again can make the analysis a bit simpler. You do one set of clustering, you've got one set of cluster labels rather than a separate set of cluster labels for different individual samples. Okay, so here's a summary of all the samples. So there are 13 samples here in total, but more than that, if you can count the number of little grids. And that's because some samples actually run across multiple different combinations of technology. So for example, we can see, what is it, sample 708, there's 2 instances of that one. 709, there's 4, 5 instances of 709. So these are bits that are coming from the same individual mouse, and then they get just gone across different sample preparation and protocols for preparing the data. So yeah, we get variation, you can see there in terms of the counts per spot, particularly the early experiments towards the top had slightly lower counts than the later ones per region. But anyway, looking at data at this level, particularly the later samples prepared with CytAssist, the biologists are like, well, I can already see that's a beautiful spleen, which is like, okay, that's great. Maybe we need to dig a bit deeper though, to actually look at which genes are expressed in some of these regions, because it does look splodgy, but again, are the splodges biologically meaningful ones? So if we look at the UMI counts across all of these different samples and data sets, and here we've got them categorized by fresh frozen, FFPE, with and without CytAssist. You can see there that there is some variation between samples individually. And also for the later data sets, which have used the CytAssist platform, we tend to be getting more data. The distribution is shifted north. So basically we're getting more signal per unit of measure. And surprisingly, that also holds irrespective of whether you've just freshly frozen or FFP preserved the tissue. So that's really encouraging. That and I guess our take home from this, what have we got here? And that's similarly covered if we look, instead of total UMI counts, the number of genes detected per spot as well. Again, we're getting more spots with more features detected in the CytAssist prepared samples compared to the fresh frozen FFP without CytAssist. So that's encouraging. It means the extra box that 10x are selling for this particular type of tissue is working great. We're getting better data. And in this final panel here showing more of the data happens to be appearing in spots under tissue. So basically, we're getting more usable data from these CytAssist prepared samples. So that's great. The next bit is to go a bit deeper. So that's just looking at sort of library size or number of features. Are we finding biologically meaningful features in these data sets? And what is the overlap between hypervariable and spatially variable genes? So we looked at this in a few different ways. Interestingly, the top hypervariable and spatially variable gene is the same, and that's a gene called CAR2. And it's really quite specific, it's an erythrocyte marker, and it's sort of specific to the red pulp regions of these spleens. So these are kind of the intermediate bits. And then you can see other expression of markers like CD74 is kind of more internal bit associated with different immune cells. And again, if you run these spatially variable genes that you get through a gene enrichment analysis using gene ontology, you can find lots of meaningful categories of like immune responses, B cell activation, which is exactly what's going on in these spleens. So that's very reassuring. If we look at the spatially variable genes recovered by NNSVG, so we're getting a single list of spatially variable genes, you can see here that the three methods, FFP, FFP cytosist, and fresh frozen cytosist, have the greatest level of overlap. And these are all probe-based methods. So again, that's suggesting that, and if we zoom in and actually look at for spleens prepared using FFPE, we're seeing much more concordance between the spatially variable genes detected for that condition compared to spleens prepared by fresh frozen methods. And the major difference between fresh frozen here is that one platform uses probes, it's the cytosist, and the other does not. And so you can kind of see that the genes and the features you're recovering are perhaps a bit different for the poly-A-based method, which is not unsurprising because, yeah, it's perhaps gives you the ability to, see other things that are not covered by probes. So there's some unique content there. The next part of this analysis is to do some clustering and then try and annotate those clusters. So we can see a UMAP of our data, and we've used ISC-MEB to cluster the data. And so we've got 7 major clusters here, and we're doing this for the cytosist samples. So for this analysis, we're focusing in on a particular subset of samples that we saw gave the highest quality data, or relatively high quality data, and just do a complete analysis of these data. So we do some clustering. We then use marker genes that we have. So a small list of marker genes for the main cell types that are present in these spleens, to then try and annotate the clusters or the groups of spots that we have into whether they are more likely to be germinal center enriched, neutrophil enriched, erythrocyte enriched, plasma cells, T cells, so on and so forth. So the heat map you're seeing there basically gives a log fold change enrichment value for these marker genes. And you can see here, you can annotate cluster 3 as germinal center enriched, cluster 4 as neutrophil enriched, and so on and so forth. So that's already very informative. We are seeing the right types of cells in these spleens. And then when we plot these results, we can kind of see about the tissue architecture. So indeed, you've got little blue spots throughout the tissue. These are the germinal centers, surrounded by clusters of B cells with patches of T cells nearby those, just like appeared in the original image. And so, yeah, and the biologist collaborator here is like, oh, well, these look like beautiful spleens, but they're not as well organized as they should be because we've treated them with malaria, and that causes, even though the germinal centers are activated, some disorganization of other bits and pieces. And again, this is like, well, this is beautiful, just like you'd expect to see with a microscope. I can see all these regions, and biologically, these look like sound spleens that we're getting good data from our Visium. So that's one level of ground truth. Do we reconstruct the different zones and areas we expect in our tissue? So the answer there is yes. But another layer on this is that, remember, I said that we have samples that are coming from male and female mice. So in those instances, a germinal center cluster should be pretty similar. But again, there'll be certain genes that are missing in one or present in the other if we focus on sex-specific genes on the X and Y chromosomes, for example. So the analysis here is to kind of go through cluster by cluster that we've annotated, and then to just run a pseudo bulk differential expression across the aggregate spot counts per cluster. And then in this case, we've got a small number of males and a small number of females in each case. So here we go, 2 male samples and 4 female samples. again, we can annotate them using the main cell type labels, cluster region type labels that we had before. So B cell, germinal center, and so on and so forth. If we then just choose a cluster type, so in this case, we're choosing the B cell cluster, and we are looking for differential expression here. You can see very clearly the Y chromosome genes are up in the males, as you'd expect. They're present there and absent in the females. And some X escape-related genes are more abundant in the female samples and thus are downregulated in this comparison. And so if you look at the ranks of those genes in your differential expression analysis, they are indeed where they should be. The Y genes are up and the X genes tend to be down. And of course, this is something that should be common across all the different clusters that we have in our data set, because those chromosomes, the expression of those genes should be consistent across the male and female samples. And indeed, that's what you see. So they're enriched systematically in the right direction with very small enrichment p-values there. And again, the set size varies slightly depending on the expression, whether a gene is expressed or not in that. So that's one source of independent ground truth. The second source, the final source, is some of these spleens had a gene knocked out. And so the collaborators had studied this gene knockout in detail in other early experiments where they'd been able to sort germinal center enriched B cells from these spleens and then do their differential analysis there on a sort of bulk manner. So they have their favorite gene signature from that earlier analysis. And so the question here is, can we overlay that on our data and check for expression of that signature in the relevant place? So in this case, the relevant place is the B cell cluster because the gene is conditionally knocked out in B cells. Again, we only have two knockout and two control. I think these are all male samples, so they're all controlled for biological sex. If we look, again, we can do a pseudo bulk differential expression analysis. TBET, which is a gene that's knocked out, is the most highly downregulated gene in this analysis. It's number one in your list. And you can see that the downregulated TBET genes tend to be a little bit below the 0 line, and the upregulated TBET regulated genes tend to be a little bit above the line. And again, we can formalize that kind of gene signature and check, are the up, and the down, or do they tend to be? And indeed, that checks out. So the roast p-value for enrichment there is 0.005. So again, this reads out pretty nicely. Oops. So yeah, that's the first benchmarking study. The data is publicly available, so you can feel free to download it and use it. There's plenty of code, I guess, with the gene signatures and all that sort of ground truth information available on GitHub, and there's a preprint available. So if you want to read more, you can get a sense of all the other results in there that I haven't had time to talk about. And yeah, so I guess, although best laid plans, we hope to generate data across lots of different platforms, but in the time and budget available to us, we're able to create a fairly deep resource for 10x Visium on mouse spleen. So if immune tissues are your favorite, this might be a useful benchmarking resource to use. So I guess what we found was that the probe-based data collection methods with the cytosist gave the best quality data. We were able to find expected immune responses, including germinal centers in our spleens, which is consistent with what you'd expect given the model that we're using. And there was, very clear differential expression signals, especially for male versus female in sex-specific genes, and also for the knockout control samples in this data set. So what I really like about this data is that we've tried to make a kind of replicate collection where we've got biological replicates in our different groups, and tried to work up how would you go through that and make a sensible analysis using existing tools. And as I mentioned, there's NNSVG and ISC-MEB have kind of multi-sample modes that you can turn on to say, right, I've got a bunch of samples here and a bunch of samples here. So it might be useful to analyze them as a whole rather than as an individual sample, which is what some methods rely on. Our ambition in the future is to expand this to other platforms if we can. Still a work in progress, and maybe add Visium HD data, which I guess is just a very new platform, probe-based, much higher resolution than the spot-based technology that's generated here is, and of course expand the benchmarking to include other analysis tools. At the moment, we've just taken a single path through the data, but you could imagine there might be multiple paths depending on how you cluster, how you annotate, if you do deconvolution or not, how you select your variable genes to use as features in your analysis. So, we're almost out of time. Well, thanks to everyone who did that work, particularly May and Chongqing, who did all the analysis under the supervision of Charity, and Danny in the lab, who supervised all the ordering of experiments and organizing the tissue to go from here to there and everywhere else. And everyone else on the respective genomics team run by Rory Bowden and Diana Hansen's our biological collaborator from Monash, who provided all the samples and all the biological insights that we can explore further to answer other questions that she has about these data. And various funders, and of course the support teams from BGI, 10x Genomics and Visium. Okay, so I'll move on to the second topic, another benchmarking data set. Why on earth would you need another one? Well, so this one, the goal is to go much broader across different technologies, and particularly sequencing-based spatial transcriptomic technologies. So this study, cadasSTre, is named after, it's an Italian term, so cadastral map is basically a little map where you know how much land everyone owns and where it all is, and then you use that to levy taxes on people. So the idea is here, well, ideally we know a little bit about the biology and the little spatial regions and how much space it takes up. And again, we'll have some ground truth to overlay on top of that to do our benchmarking. So I did my PhD on microarray. We're back here. Microarrays are still useful, and now they're useful for probing spatial transcriptomics. So for example, we've got a range of different ways of profiling that are listed on this slide. Microarray is one of them, a popular one. 10x Visium uses this approach. And of course, you've got spots of different sizes. You can also have beads or polonies or nanoballs on your surface, or you can use microfluidics. And so for all of these different approaches, at the end, you've got to have the tissue come in contact with the surface that has these different ways of profiling gene expression. You've got to make a library, and then of course you're going to get your data matrix and do some sensible inference on it. So there's a lot of data processing to happen. So a challenge here, if you're going to use more than one technology, is they all have slightly different spatial resolution. So from stereo-seq, which are lots of tiny little DNA nanoballs, up to Visium, which are pretty large regions. And so it's a bit different to a bulk or a single cell benchmark experiment where the unit of measurement is a cell. Here it's like, it actually depends. The unit of measurement varies. Another issue that I guess I touched on before is, well, what is the relevant reference sample to use? So, cell density can vary. So in spleen, it's really densely packed. In other tissue, it's a bit more spread out. So there's lots of variables depending on the tissue that you take. And yeah, different cell types can vary across development, and there's can be all kinds of things. So the idea here, once again, is let's use a reference tissue that we can get relatively easily again and again. We're going to brain or mouse embryonic eye, the adult mouse brain, or the mouse olfactory bulb. So 3 different tissue types. And then 11 different sequencing-based spatial technologies, from Visium to slide-seq to stereo-seq and dbit-seq, covering microfluidic system. And then also have some reference 10x data to match across to. And the data analysis pipeline here, again, from raw data, we are going to focus on particular regions that we can sort of work out where the relevant region is using in situ hybridization data on these sections of tissue, generate a count matrix, and then do downstream analysis with deconvolution, clustering, and differential analysis, and so on and so forth. So this is a very ambitious study, which has data across. So here we're starting with the mouse embryo eye. So we're not just capturing the eye, we're capturing regions around the eye. Some technologies that we have to be quite focused because the region that you capture is small. Others, we're kind of capturing a lot of space around it as well. And you can see for scale there's a bar that varies in each little picture, depending on the resolution that we have. across the different technologies. So mouse embryo eye, and what we're showing here are UMI counts across these different regions, and you can see they vary enormously depending on the technology. Next is the adult mouse brain, of which the hippocampus is a subset of that. So that's kind of the squiggly bit as a hippocampus, and then you can get varying regions around it depending on the amount of space you have to profile on your chip. So again, we've got UMI counts. They vary between technologies, and yeah, but in each case you can see the little squiggle, which is the region that we're interested in. And finally, mouse olfactory bulbs. So you might have one, you might have two, but they are looking quite characteristic and similar across these different platforms also. So we have quite a large collection of data, and the challenge here is, well, how do you kind of control for variations in sequencing level, variation in resolution, all of that sort of stuff. So the approach here is to hone in on the particular region of interest. So in this case, this example here is a mouse hippocampus. We can obviously take all the data that we get according to this defined region in terms of, so the x-axis on this middle picture is the amount of sequencing reads, and the y-axis is the total counts that we are actually taking into account for this region. You can see there's quite a range of sort of effective, there's a large variation in the amount of sequencing data we have, and there's also a large variation in the amount of data we use that makes it into our data matrix. And so here we go. We can see if we categorize by all reads versus downsampled here. The downsampling approach is to try and control for this. So basically we take, it's kind of limited by the technology that gives you the least data. So you have to go, right, what was the kind of that gives us the rate, the number of reads we can use. And then we downsample here. And you can see, so for an example, this example here, we've got SlideSeq v2 actually gives you the most counts for the hippocampus, and then the other methods follow off there in the different colors. So we're following by Visium, and then what have we got there? Another slide, or it's Curio, and so on and so forth. So this is for the mouse hippocampus. Again, for the eye, the amount of tissue we get around the eye varies, but if we focus in on just the eye region, we could again make this analysis and do downsampling and see effectively, how much usable data do you get if you try and account for sequencing depth variation in these tissues. So in this case, the Visium data, probe-based tends to do better, and then we followed up by Dynaspatial and then SlideSeq again. So this is total counts. You can then go and pick out particular marker genes and see, look, are we detecting these across these different spatial platforms? And so you can see there for all key marker genes for the hippocampus on the left and the mouse eye on the right, we do indeed detect some reads for these markers in the region of interest. So that's, again, showing signal, not noise. So that's great. Another thing to note is, as you might hope, with spatial transcriptomics, you hope you can find things that might be missing in other data sets that you've analyzed up to this point. So we've got some clustering results here for 4 technologies: StereoSeq, Visium, PMK, SlideSeq version 2. And the clustering here has been annotated. So we can see in this developing embryonic eye, we've got the blue regions, which are lens, annotated as lens, regions around it. And so quite nice structure that all of these technologies are recapitulating from our clustering analysis. If you take particular markers of these different regions, so, PMEL is a marker of melanocytes, or we've got CRYBB3, which is a marker of the lens, you can again see, right, this looks to be spatially variable, quite concentrated. And again, you've got different markers, which, again, are distinguishing different bits of tissue. So if we go to our reference data, which is just regular single cell on these tissues, which again I've tried to enrich for the eye region, you can see that although you can find markers for the purple and blue regions of the tissue in the single cell data, If we look for markers of the lens, for example, there's not really any clusters recovered for that one in our single cell data. And I think similarly for the red, we don't have any clusters that are really enriched for those markers either. So we're definitely getting extra bits of information that we might have missed before if we're just restricting ourselves to developing atlases for based on single cell data. In this case, it's nuclei because of the region that we're looking at. And yeah, in some technologies, particular marker genes are missed. So that's another thing to be aware of. So we've got these genes that start with CRY and a few others, which are specific again for the lens. These genes light up nicely in the stereo-seq data, but are a bit randomly expressed in the poly-A-based Visium data. And you can see how basically all of these markers, the expression is kind of missing in the Visium poly-A-based data. But if you make a probe for these genes, so the third row there, we can again see we can pick up these as you expect. So I guess that again suggests the way 10x is going towards probes, and there's maybe a good reason for that because you can really focus in on regions and not run the risk of missing particular genes in regions of interest altogether. The final bit of analysis is about diffusion. So again, if we use histology to try and pick out a particular region that has certain marker gene expressed, we can try and align the tissue across different platforms and rescale it and then check, well, how distinct is the capture for that region where, again, the in situ says, no, this gene is really distinctly expressed exactly here, versus how well does the spatial data recover that? And so you can see there's a bit of variation in terms of the kind of drift of RNA, sort of left, right of that center peak that varies a bit between technology. So this is for olfactory bulb. This is for hippocampus. Again, there's maybe a bit more drift in the hippocampus, particularly for stereo-seq and the third technology there. In a different tissue, perfectly sharp. So again, there's this sort of diffusion effect that's a bit variable between tissue. That's maybe something to be aware of when analyzing this sort of data as well. And here's a measure of this metric that, again, there's no consistently more diffuse technology that has more diffusion than others. It sort of varies tissue to tissue. Okay, so in the last 30 seconds, this is a nice summary of this analysis. I implore you to read more if you would like. There's a preprint. It's a QR code. It will take you there. The data is available online, and there's a really detailed sectioning protocol as well, which is helpful for, again, making this sort of data collection reproducible between different labs. So yeah, diffusion is something to be aware of. There's definitely more detail in these spatial data sets, as you would hope, that might be missed in earlier single cell atlases. Certain biases do exist depending on the flavor of Visium. And yeah, your sequencing might need to be a bit more deep than you. So for example, in all of these data sets here, we never actually saw saturation. So if you kind of sequence more, you're still finding more genes that are adding to your total UMI counts in that analysis. And again, there's the goal to update this data set over time and add more technologies. So the final perspectives here is, well, hopefully the data could be useful to people in the room. So if you have an interest in benchmarking, then maybe some of these data sets that I mentioned, cadasSTre and SpatialBench, might be something to use or reuse. Another project you might be interested in knowing about is SpaceTX, which also has like reference tissues that they've tried to generate data from across multiple different technologies. The field is moving quickly though, so the shelf life of these data sets might be quite short. Maybe all of these data sets are obsolete very soon. I guess maybe those Visium technologies won't be available forever. I guess stereo-seq are always releasing new versions. And again, more commercial companies are coming on board with, version 1 and then version 2 and lots of different versions. So it's a challenging field to keep up with, but I guess we need to start somewhere. So hopefully this data is a start. And yeah, there's definitely, I think, a need for having sort of more living benchmarks where rather than you do your analysis, you publish your paper, is it possible to make the data available and shareable and, the analysis recomputable so that as new versions of people's software come out, you can again check that they perform as they did before, or maybe ideally they perform better than they did previously. definitely reach out if you're interested in collaborating on any of this, or you can just download the data yourself. So feel free to do that. So this project was led by Luyi Qian, who was a former PhD student who now runs his own lab at Guangzhou Laboratory in China. And Yue was also a former PhD student who's the first author of his study. So well done, Yue, on your first postdoc preprint and hopefully publication soon. The data generation was led by Ethan Liu, who runs a lab at Westlake University, and Yuting Fu is a person leading that work. And then I just wanted to highlight Yang Su, who's a former University of Sydney student who just started a PhD in the lab, who'll continue some of this benchmarking work. I think he used to work with Pengyi. So maybe he can give a talk in a future Zoom meeting here. So thanks, everyone.